External Structures, Chapter 3, Topic 3.2. In this topic, we will be discussing the external structures of prokaryotes. We're going to talk about what these external structures are. We're going to talk about the glycocalyx, flagella, and structures for attachment and mating. At the end of this recorded topic lecture, you should be able to meet the following objectives. Describe the structure and function of 4 different types of bacterial appendages and explain how the flagellum works in the presence of an attractant. Appendages in prokaryotes are not arms and legs, that kind of thing. They are accessory structures that not all bacterial species have, and they perform 2 main functions. And those are motility, which is movement of the cell through its environment, and attachment, which is grabbing a hold of things in the environment. So all of these accessory structures extend outside the cell of the bacteria or the archaea.
Glycocalyx means sugar cup, and it refers to the glycocalyx being composed mainly of carbohydrates. Now, the glycocalyx performs 2 functions: preventing the cell from being eaten by amoeba-like cells, and 2, helping it stick to stuff. Now, sometimes both forms of the glycocalyx can perform both functions, but for the purpose of this class, we're going to say that they're 2 separate functions. What in the world am I talking about? Well, if you look at this micrograph of these bacteria that have a glycocalyx around them, and that's the white, clear area around the dark cells, the glycocalyx in this situation is very tight around the cells. It kind of wraps them in a slippery cocoon. We call this a capsule, and it prevents the bacteria from being eaten by other amoeba-like cells, or the fancy term for that is phagocytized. Generally speaking, if this is a pathogen, they are more pathogenic if they have a capsule, because it prevents your white blood cells from phagocytizing them, from eating them down. Now, the other form of the glycocalyx is the slime layer. That's where the carbohydrates are less attached, less closely attached to the cell, and they have a tendency to be more sticky. And its function of the slime layer is to stick to other cells and to stick to other surfaces. They have a prime function in forming biofilms. So in the morning when you wake up with tooth fuzz, okay, all of those bacteria that have been dividing throughout the night, they have been producing a slime layer that allows them to attach to each other and to your teeth. By the way, you might want to break out a toothbrush and bust up that lovely biofilm. Flagella are hair-like structures that stick out from the bacterial cell and allow the bacterium to swim around. Okay, you can see the flagellum here. Flagellum is singular, by the way. Now, if the flagella comes just out of one end and there's just one of them, we call this monotrichous. Trichous is Greek, I believe, or Latin, for hair. So it's mono-haired. Now, if you have a cluster coming out of one end, we call that lophotrichous. Now, if they're coming out of both ends, now we call it ampitrichous. Ampi, if you're ambidextrous, you use both hands equally. So this one has flagella coming out of both ends. And you see that on Spirillum more often than not. And then if you have flagella all over the place, you are peritrichous. Let's talk about the structure and movement of flagella. Now you're probably wondering, why bother? Well, it becomes important later on when we talk about the difference between prokaryotic flagella and eukaryotic flagella. Now, prokaryotic flagella are composed of a molecule of protein called, well, it's a protein, which is a molecule, and it's called flagellin. That should make it easier to remember. And it's stiff, and it comes out of the cell wall. It's embedded in the cell membrane, goes through the cell wall, comes out, and it's set up with these discs. so that the flagella can turn 360 like a little outboard motor. Later on, I'm going to have a little film of swimming bacteria that you can see the flagella spinning. Now, later on, we're going to talk about the difference between gram-positive and gram-negative bacterium. But if you look over here, the one to the left is gram-positive. It's got just one thick peptidoglycan cell wall. Gram-negatives, because they have two membranes, one thin layer of peptidoglycan, they're a little bit more complicated, but they still spin 360, and they're composed of flagellin. Spirochetes have a specific structure to their flagella that are called endoflagella. They're kind of like ingrown hairs. What happens is the flagella come out of the cell wall, but then they come back underneath an external sheath. It's kind of like a capsule. You'll notice on this micrograph you can see where they have both, where we've got both flagella meeting. You can see two of them. You can see just one right here. And we call this endoflagella. What happens is, when the flagellum spins, instead of spinning just the flagellum, spins the whole cell. So when you look at it underneath the microscope, they go spinning through the medium like little corkscrews. And this is seen only in spirochetes.
Now let's discuss how a flagellum functions. Suppose in the container that I have my bacteria, I have dropped some food. We'll say this is a sugar cube. This is a huge sugar cube though. Anyway, so as the sugar cube dissolves, molecules of sugar are going to be in higher concentration here, even less. So my bacterium is swimming around and it realizes that there's food, so it changes direction, and it realizes that it's getting further away from the food because the concentration of sugar molecules is less. So it's going to do what's called a tumble. It just kind of spins around randomly, in the hopes that when it starts off in the new direction, it's going to be getting closer to the food source or the higher concentration of food. So when it's going in a straight line purposefully, we call this a run. Well, it can tell that it's getting further away from the food source, so it's going to do another tumble, and this time the run got it going in the right direction to go toward the food. We call this chemotaxis. Chemo for chemical, taxis for movement. So this is how prokaryotes move toward chemicals or away from them. Let's talk about that. In this example, I've given you an example of positive chemotaxis, or the bacterium is moving towards something it wants. It can do the same thing in moving away from something it doesn't want. Suppose I put in a toxin in here. It's going to do mainly tumbles until it can get away. And we call this negative chemotaxis.
Pili are both composed of a protein called pillin. That makes it easier to remember. And it used to be that the two terms, fimbriae and pili, were used interchangeably. Now they're separated, mainly based upon what they do. Fimbriae are used to attach to other cells and to surfaces. They can kind of fulfill the same purpose as a glycocalyx. Pili, on the other hand, are specific for grabbing a hold of other bacteria for the purpose of exchanging genetic material.
Okay, that's it for this topic. You should now be able to describe the structure and function of 4 different types of bacterial appendages and explain how a flagellum works in the presence of an attractant.